Okay, so the MVIC test is another one that you'll be expected to know. So the purpose of this test is multifold, so that it's obviously not a single test. This is the series that's used, but what is the use for the MVIC series? So there are two organisms that it differentiates between. Does anyone remember the organisms? Okay, E. coli.
Enterobacter, something?
Which Enterobacter?
Aerogenes?
Aerogenes, okay, good. And so the reagents used, I'll have a chart for this. I'm not going to ask everyone to say them right now. But since there are a lot of tests, there are a lot of indicators, a lot of media to remember. So it's useful to put this in a chart. And for E. coli and Aerogenes, you do want to know which organism produces which set of results for each test. So indole production is done by a tryptophan tube. So that one is harder to remember with the mnemonic because tryptophan doesn't start with an I, but that's the indole production. Methyl red is fairly easy, Vogts-Proskauer and citrate utilization. So a lot of you used at least one or two of these tests somewhere in your unknowns, if not looking at indole directly by tryptophan, looking at indole through the sim tubes or something like that. So just be able to recognize these tests and what they do and who they're trying to differentiate. So here's an example. We have indole, methyl red, Vogts-Proskauer, and citrate. So for the tryptophan broth, what would the observation be? You probably can't see that in the back, huh? It's too small. It's red. So that would just be the indicator turning red for tryptophan. What about methyl red?
It's red.
It's red. So methyl red, if you remember, the media starts yellow, and when you add the indicator, if it's positive, it turns red. Vogts-Proskauer, I'll give you a clue. So methyl red and Vogts-Proskauer are done with the same media. That you just split the tubes and use a different indicator.
No change.
No change. So that's what the media looks like initially. And what about citrate?
Green?
Green, exactly. So green or no change I would also accept because the green is the initial color. So what about the interpretations? This one's a little bit more, there's a little more to it. So for the tryptophan, if you have red, what does that indicate? Right, so that indicates indole production. So you don't want to just say tryptophan positive. Indole positive would be okay, but you can't always just say the media and then positive or negative. You want to know what it actually represents. So a good example of that is for methyl red. So what would the interpretation for methyl red be? It's not just methyl red positive. That would be a partial credit, because at least you knew that red was positive and yellow was negative. Right, so red on methyl red indicates mixed acid fermentation. What about for Vogts-Proskauer? Does anyone remember what that represents? All right, I'll bring it up. What about citrate?
Citrate utilization.
So it was negative for citrate, so you can just write citrate negative, or it does not utilize citrate. So, as I said, for methyl red positive, you do want to say that this is mixed acid fermentation. I'll accept methyl red positive, but only for partial credit. And the Vogts-Proskauer was 2,3-butane-diol. This is another one where if you say VP positive or VP negative, that's only worth partial credit. You need to say what it actually is looking at. So what is the biochemical meaning of that result? So just as another example here, this is actually representative of either E. coli or Aerogenes. So who is it? E. coli. Good job. So Aerogenes has the exact opposite results. So another one that we're looking at is the carbohydrate fermentation tubes. So what's the purpose of this media? What do we use it for?
Test for carbohydrate fermentation.
Right, so for each carbohydrate that you're looking at, it just tests whether or not the organism can utilize it. And the indicators, I won't go through them all right here. For fermentation, it's rum-cresol blue, and that's for all the tubes. That's just the color that we use. It's just a pH indicator. And for gas, remember that these tubes have a Durham tube inverted, and so if gas is produced, it'll collect inside the Durham tube. So the media results, For these, I won't go through all the observations and interpretations because it'll get tedious, but all these will be posted online so you can go back and look. But this is another example of where you can get tripped up by only providing a partial interpretation. So if you just say for A, if this is glucose, if you just say glucose negative, that's only part of what you're looking at. You want to say that glucose is not fermented and that no gas is produced. So you have to remember that you're looking at both fermentation and gas production. And for the observations as well, if you don't mention the bubble in the Durham tube or the lack thereof, that's still partial credit.
Does it ever produce gas without fermenting the sugar?
I've never seen that happen. At least it's not supposed to happen under our settings.
Okay.
Okay, so what about urease and phenylalanine? I think these are two of the last media. There might be one more that we want to go over. So urease is pretty straightforward, right? Test for whether an organism possesses a urease or not. Phenylalanine is looking for the enzyme phenylalanine deaminase. So those are ones to remember. Urease is easy. The phenylalanine takes a little bit more to remember because the purpose and the enzyme you're looking for isn't directly in the name of the media. And for the media results here, if you remember, urease starts off as orange, and so if it turns pink, that's positive. So you can say urease positive or possesses urease, either of those is fine. And for phenylalanine deaminase, you want to make sure that you say it contains phenylalanine deaminase or is phenylalanine deaminase positive. If you just say it's phenylalanine positive, then that's only partial credit. So this one turns yellow to green. So you will also be tested on the unknowns in sort of a broad sense. Things like the media that was used, we just went over a lot of that. But you should be familiar with most of the media that you've used throughout the unknowns. And why do we use controls? This is probably one of the biggest sticking points for the practical. So what makes something a good or a bad control for a given test? And these are just rhetorical questions at the moment. So that depends on the test, obviously, right? If you're doing something like a TSI, especially for the purpose of this class, you can't do a control for every single possible outcome. That's just too many tests to try. So you just want to do enough controls that you know that the media is working. But as far as being good or bad controls for something like EMB, on that it's a plate. You can divide it up really easily. You can say, I know E. coli and Citrobacter fruendii both produce that metallic green. I know this organism produces pink, this organism is opaque. So you can use all those different controls. So you want to make sure the media is working first and foremost. And you also want to make sure that you have enough controls to cover your bases as far as the possible results when that's something that you can do easily. And then for the use of controls, what can we conclude about an experiment if the controls fail? And this is just rhetorical again. What if they're not run? Is the experiment valid? Do you need to redo it? Or if the controls fail, do you need to redo the experiment? And if the controls are successful, then what can you conclude about your results? So this is mostly, the unknown questions are going to be focused on the importance of controls and their utilization. So this is just an example question. If you inoculate MSA plates with your gram-positive unknown and observe after 24 hours, you see no color change, can you be confident in this result? And if not, what should be done differently? So does anybody have an answer?
You don't have any controls, then you can't accommodate it.
Right, so you, especially when there's no color change, you don't know if the media worked at all. Maybe they didn't add the pH indicator in the first place, and so you're just streaking out on red-colored plates that don't do anything. So this is another one where you need to have the controls, and so that was the right answer. So we'll also be looking at water testing and coliforms. So you already had a question like this on one of your quizzes. What traits differentiate coliforms from other organisms? There were a list of possible traits that tell you something is a coliform, so you want to know those. And what media tests are used to look for coliforms? So for this, you want to know the full names.